In this third and final portion of our unit on the vestibular system, we're going to talk about the vestibulo-ocular reflex, which functions mainly to compensate for head movements in helping to produce a stable image on the retina, so that our vision will be able to continually guide stable positions and stable movements. And as an example of how this works, if you place a book or a piece of paper in front of you, and as you're reading a page in that book, and you rotate your head to the right, you'll still be able to read the words in such a way that your eyes will rotate towards in the left direction as your head rotates to the right, in a manner to match the speeds of the rotation to maintain the stability of that retinal image. Now, as an example, if you don't rotate your head and you move the book, you'll find it much more difficult to read those pages, because the vestibular system is not activated. When your head is stable, you're not discharging action potentials to the vestibular nuclei. All you're doing is generating impulses through the optic nerve. And our visual processing is slower than our vestibular. So when you actually move the book and not move your head, it becomes much more difficult to read those pages, because you're not getting simultaneous input from your vision and the vestibular system. There are three basic reflexes involved in the vestibulo-ocular reflex: rotational, translational, and ocular counter-rolling, which is what we'll talk about here in this third and final segment. In terms of rotational, as the term suggests, it deals with head rotations, and so the semicircular canals are specifically designed to detect these types of movements, and they will generate eye rotation in the opposite direction. So using the same example we had on the previous slide, if you have a book in front of you and you rotate your head while keeping the book stable, you're still able to read the pages, because as your head rotates to the right, your vestibulo-ocular reflex will trigger your eye muscles to rotate to the left. And there are two phases for this. There's the vestibular, what's called the slow phase. It's certainly not slow, but with respect to the second phase, it is considered relatively slow. And this is this results in the movement of the eyes that's opposite to the movement of the head. And it's referred to as slow because it is limited by the orbital range, and so if we rotate our head too far, that will trigger this reflex to readjust, and that's what's called the quick phase. And so our eyes will then displace to a new point of fixation that's much faster than the vestibular slow phase in the direction of head rotation. So if our body is spinning in the right direction, the vestibulo-ocular reflex will then generate a leftward rotation of our eyes. When we approach the orbital range, the quick phase will then, in a much faster manner, rotate our eyes to the right in the direction of our rotation, of our body rotation. Will fixate on a new point in space, and then the ocular reflex will then reestablish itself in the slow phase. And this illustration demonstrates this process. And so in this particular example, the head is turning to the left, and as it turns to the left, we see that the stereocilia are rotating in the direction of the kinocilia, and as they're pushed in that direction, we see activation of the kinocilia, of the hair cells. And those hair cells will then discharge action potentials through, eventually through the vestibular nerve, and that will increase the rate of firing. On the right side of the body, the rotation that is generated will cause a bending of the stereocilia away from the kinocilia, and that results in a hyperpolarization of the axons that project from these that project out of the semicircular canals, that will eventually control our eyes to compensate for that rotation of the head in the left direction. So that causes a decrease in the firing, and that's the rotational component of this reflex. Now, when we talk about linear motion and head deviations, these are primarily sensed by the otolithic organs, and this is really responsible to allowing us to compensate for the distance that objects are to us. So closer, and that's why closer images tend to move faster across the retina than farther images. And so this linear motion and head deviation detection is much more sensitive to closer objects than it is for farther objects. In terms of head tilts, we think about movement of the head away from a vertical line, and so eyes will tend to rotate in the opposite direction as an attempt to reduce the tilt of the image. And so as part of the vestibulo-ocular reflex, the objective here is to maintain a stable image on the retina. These reflexes are typically effective with the onset of movement or an abrupt change in movement, but compensations are typically poor with constant speed motion, whether it's translation or rotation. As discussed in in the first part of this unit, the vestibular organs are particularly sensitive to accelerations because it's the accelerations that induce forces contained within the hair cells. And so getting up to a particular speed that is constant will see a reduction in the discharge of those vestibular axons. And so low linear accelerations, very slow rotations, will see a reduced influence of these reflexes. So our ability to compensate for movements tends to be quite low. Now, how do we relate this? How do these neurons react with when we talk about combined inputs from two systems? And in this particular set of experiments, we see researchers examine the medial vestibular nucleus, and they recorded the activity of neurons coming out of this particular cluster of neurons. And they translated this, or they measured the activity in terms of the frequency of discharge. And so in this experiment, animals were rotated in the dark. So when they were rotated in the dark, of course, visual information cannot be incorporated into the central nervous system, particularly here into the vestibular nuclei. And so what we see here with body rotation in the dark, we see an initial spike in the discharge frequency. So we can see here, here we see a stable, no rotation condition on the kinematics. We see an acceleration period to a constant speed of rotation, and then back down to a decelerated position to a resting state again. And so we have the tonic discharge of these neurons, a steep spike that corresponds to the acceleration phase, and a decrease and accommodation of those discharges from the vestibular or the medial vestibular nuclei back to the point where they will continue all the way to zero when the acceleration is in the opposite direction. And so this accommodation takes place during continual rotation. But when we rotate the animal in a lighted condition so that visual information is provided, then what we see here is we see a maintenance of discharge from the medial vestibular nucleus. And so not only are vestibular nuclei discharged, but information coming in from visual sources will be additive to the output of the medial vestibular nucleus. Interestingly here, what we see here is that the animal was not rotated. However, the visual scene around the animal rotated to simulate body rotation. So in this particular instance, we have isolated ocular stimulated visual senses, but we don't have any vestibular activation since the body did not rotate. And so in this particular case, we see that initial spike of activity of the medial vestibular nucleus maintained at a steady state, and then back down to that tonic level. However, the onset of the activity of these nuclei occurred slightly later than what was observed earlier in the previous two experiments, suggesting that the visual system is a little bit slower than the vestibular system. So when the vestibular system is activated, it responds much quicker than if we were to rely purely on visual input. In terms of cerebral cortex integration, these, in terms of the projections to the cerebral cortex, they all, of course, go through the thalamus, through the ventral posterior and the lateral portions of the thalamus. Synaptic connections onto relay nuclei will then terminate into what's described as S1, or the primary somatosensory cortex. So visual and somatosensory inputs will also terminate in this region. There are reciprocal connections between the visual and the vestibular centers, and those reciprocal connections between those centers helps to resolve contradictory information. And so in the previous slide, when we had the animal responding to or examining the response of the animal to a rotating visual scene, that can result in a disequilibrium type of condition when we don't have that mismatch, or when we have a mismatch between those different sensory systems. One example here is riding in a car. So when a person is driving in a car, we're able to correctly perceive self-motion as the car accelerates, moves around turns, achieves a steady state, so on and so forth. Understanding that when a car has achieved a constant speed, there is relatively little vestibular activity. However, our vestibular system is quite sensitive to very small accelerations that take place as the car rides along the road. And in this particular, these two examples here on the left-hand side, here we see a person driving a vehicle, although his hands are not on the wheel. They should be, but here we see somebody driving along the road. Here we see somebody in a driving simulator. And the difference between these two conditions here is that this person, despite that they may be moving at a constant speed, is still generating discharges through his vestibular apparatus. If he were city driving, the accelerations and decelerations that take place with city driving and moving around turns will provide input to a central nervous system that should coincide with his visual input. In this particular case, the person is not moving. There's no somatosensory information through vibrations from the road up to the person's body, and there are no vestibular signals being generated, only purely visual signals. And so in this situation, the person may perceive or experience some degree of equilibrium, particularly if they're an experienced driver and their central nervous system is expecting input from those other systems. And this slide essentially provides a summary of how visual and vestibular signals are incorporated that are modified by the activity of the cerebellum. And so recall we have the cerebellum with a particular region that is dedicated to receiving inputs from the vestibular nucleus. And when we have a visual signal that doesn't correspond with other senses in our body, that signal will be transmitted of course, triggered by motion of the retinal image into the cerebellum, where it synapses onto the major output neuron of the cerebellum, which is the Purkinje cell. And so when we have simultaneous simultaneous vestibular information coming in through the semicircular canals that sense head rotations, that information will terminate, once again, indirectly project to the cerebellum through the mossy fibers that will alter the discharge of the Purkinje cell, which was discharged by the visual image. And by controlling the eye movements, that allows us to change the sensitivity to these muscles to the vestibular input. So in this particular case, vestibular input will help our eyes to control the muscles of our eyes to fixate on an object when the retinal image presents some type of error signal. So this ends the third and final segment of our vestibular unit. Next week in class we'll start discussing sensory motor integration.